Here are 5 USMLE style questions focused on regulation of gene expression, including concepts like promoters, enhancers, silencers, and epigenetics. Question 1. A mutation occurs in the TATA box of a gene's promoter region, disrupting the binding of transcription factors and RNA polymerase II. What is the most likely consequence of this mutation? A. Increased transcription of the gene. B. Decreased transcription of the gene. C. Increased translation of the gene product. D. No change in transcription levels of the gene. E. The gene will be silenced completely. Answer B. Decreased transcription of the gene. Explanation: the TATA box is part of the promoter region where RNA polymerase II and transcription factors bind to initiate transcription. A mutation in the TATA box impairs the binding of these factors, leading to decreased transcription of the gene. This will result in lower gene expression. Question 2. In eukaryotic cells, which of the following best describes the function of enhancers in gene regulation? A. Enhancers bind to repressor proteins to decrease gene expression. B. Enhancers are located only near the gene they regulate. C. Enhancers bind to activator proteins to increase gene expression. D. Enhancers are sequences that inhibit the binding of RNA polymerase. E. Enhancers are always located within exons of the gene they regulate. Answer: C. Enhancers bind to activator proteins to increase gene expression. Explanation: enhancers are DNA sequences where activator proteins bind, increasing the transcription of genes on the same chromosome. They can be located near, far from, or even within the gene they regulate, such as in introns. Question 3. Which of the following would most likely occur if a mutation in a silencer region of a gene disrupted the binding of repressor proteins? A. Increased gene expression due to enhanced transcription. B. Decreased gene expression due to loss of transcription factor binding. C. Gene expression would remain unchanged. D. Decreased gene expression due to loss of RNA polymerase II binding. E. Increased gene expression due to loss of transcriptional repression. Answer: E. Increased gene expression due to loss of transcriptional repression. Explanation: silencers are regions where repressor proteins bind to decrease gene expression. A mutation in the silencer that prevents repressor binding would result in increased gene expression, as the normal repression of transcription would be lost. Question 4. Which of the following mechanisms is primarily involved in epigenetic regulation of gene expression without changing the underlying DNA sequence? A. DNA mutations. B. DNA methylation, histone modification, and non-coding RNA. C. Enhancer binding by transcription factors. D. Splicing of pre-mRNA. E. Transcription factor binding to the promoter. B. DNA methylation, histone modification, and non-coding RNA. Explanation: epigenetic regulation refers to changes in gene expression that do not involve changes to the underlying DNA sequence. Key mechanisms include DNA methylation, histone modification, and the action of non-coding RNAs, which can influence the structure of chromatin and the accessibility of genes for transcription. Question 5. A scientist is studying a gene in a eukaryotic organism. The gene's expression is found to be highly regulated, and it is observed that the gene is turned off in certain conditions despite the presence of transcription factors. Which of the following elements is most likely responsible for repressing transcription in this case? A. Enhancer. B. Silencer. C. TATA box. D. Exon. E. Promoter. B. Silencer. Explanation: a silencer is a DNA element where repressor proteins bind to decrease or silence gene expression. In this case, the gene's expression is repressed despite the presence of transcription factors, suggesting that a silencer is inhibiting transcription. These questions cover